The topic under discussion delves into the intricate mechanisms of cell injury, scrutinizing both direct and indirect causes alongside cellular responses to damage. Within living organisms, various factors can incite cellular injury, resulting in either reversible or irreversible deleterious effects. Among the direct causes of cell injury is the application of heat, while indirect pathways encompass genetic derangement precipitated by metabolic disturbances and altered immune responses. Free radical injury emerges as a significant indirect mechanism, revolving around free radical formation. Free radicals are frequent byproducts of standard cellular activities such as energy generation, lipid and protein breakdown, and inflammatory processes. Molecular oxygen, O2, frequently spawns free radicals due to its two unpaired outer electrons. These radicals can detrimentally impact cell proteins. Hypoxic cell injury represents another crucial mechanism. Hypoxia leads to oxygen depletion within cells, subsequently interrupting oxidative metabolism and impeding ADP synthesis. Particularly vulnerable are brain cells, which can sustain permanent damage within 4 to 6 minutes of oxygen deprivation. Ischemia, defined as reduced blood flow due to circulatory disorders, exacerbates hypoxic injury. One of the preliminary consequences of diminished ADP is acute cellular swelling, attributed to the dysfunction of the energy-dependent Na+/K+ ADPase membrane pump. Furthermore, impaired calcium homeostasis can unleash enzymes that provoke widespread cellular damage. Such intracellular disturbances can foster reversible or irreversible cell injuries, ultimately culminating in cell death. Cell death manifests through either apoptosis or necrosis. Apoptosis signifies programmed cell death, facilitating the orderly removal and replacement of superfluous, improperly developed, or damaged cells. This process is instrumental in maintaining homeostasis within cellular populations, exemplified by the continual renewal of blood cells. Characteristics of apoptotic cell death include controlled autolysis, cell shrinkage, and membrane alterations that beckon phagocytes to engulf apoptotic fragments, ensuring inflammation is not provoked. Conversely, necrotic cell death is considered pathological, resulting from severe cell injury. Necrosis precipitates cell swelling, membrane rupture, and subsequent inflammation, divergent from the structure degradation observed with apoptosis. A significant distinction lies in necrosis exacerbation of inflammation and often obstruction of cell replacement and tissue regeneration. Shifting the focus to genetic components, the narrative explores the underpinning of genetic information within deoxyribonucleic acid, DNA, a remarkably stable macromolecule housed in cell nuclei. DNA harbors genes, fundamental units dictating protein and enzyme synthesis essential for cellular functionality. Genes serve as the hereditary currency, perpetuated through generations. Mutation-induced errors in genetic material, though often rectified by intrinsic DNA repair mechanisms, can disrupt cellular fidelity and propagation. Protein synthesis is orchestrated under the genetic oversight housed within the cell nucleus. Messenger RNA conveys coded instructions from DNA to the cytoplasm, while transfer RNA ferries appropriate amino acids. Ribosomal RNA provides the mechanism necessary for protein synthesis. Hence, the synthesis of cellular proteins underscores DNA-directed regulation of cellular activity. Genetic terminologies include genotype, denoting stored genetic information, and phenotype, describing observable traits. Intriguingly, disparate genotypes may confer identical phenotypes. In genetic disorders, expressivity defines the gene's phenotypic manifestation, while penetrance reflects a gene's ability to fulfill its functional directive. Finally, the exploration touches upon predisposing factors influencing cell injury susceptibility. Age, sex, lifestyle, environment, and heredity are highlighted as significant risk factors. Youth are susceptible to accidents, while aging increases vulnerability to conditions such as diabetes or heart disease. Conversely, gender-specific propensities manifest in disease prevalence, for instance, osteoporosis in women or gout in men. Lifestyle choices, environmental conditions, and hereditary genetic predispositions cumulatively contribute to the risk landscape for cellular injury and consequent pathologies.